On today's episode of Neural Frontiers, we are examining how artificial intelligence is transforming neuroscience drug discovery and whether the hype matches the reality. Machine learning algorithms can now predict drug target interaction and optimize molecular properties with unprecedented speed. However, the brain's complexity far exceeds the data sets these algorithms were trained on. How do we prevent AI from simply amplifying existing biases in neuroscience research? Are we at the risk of AI confidently predicting the wrong answers?
That's a really important issue. While AI can process massive data sets and draw connections, we might miss the old saying, garbage in, garbage out, still applies here. If our training data reflects biases or just doesn't capture the brain's complexity, AI could just amplify those mistakes with added confidence. What crucial is transparency, regularly validating AI predictions with real world experiments using diverse data sets and building in checks to spot biases early. Ultimately, AI should guide and accelerate human discovery, not replace careful scientific evaluation.
So, digital phenotyping through smartphones, wearables, and sensors offer continuous monitoring of neurological and psychiatric symptoms. This creates massive data sets but also introduces a lot of noise from daily life variability. So, how do we distinguish clinically meaningful signals from lifestyle noise? And could digital biomarkers actually be more sensitive than traditional clinical assessments? What do you think?
Digital phenotyping gives us an amazing window into real life behavior and symptoms that clinic visits can't capture. But with so much data, separating what's clinically meaningful from daily variability is a huge challenge. The solution lies in clever signal processing using algorithms and human oversight to filter out the noise. In some areas, digital biomarkers might even be more sensitive than traditional tools, capturing the subtle changes earlier. It's early days, but if it validated properly, these digital measures could revolutionize how we track brain disorders.
Right. So large language models trained on specific scientific literature can now generate novel hypotheses about brain function and drug mechanisms. However, these models don't understand causation, but only correlation patterns in text. Should we be concerned about AI generated hypotheses leading us down scientifically plausible but ultimately wrong paths?
Large language models are fantastic for generating new ideas rapidly. But as you said, they can't distinguish between true causation and just patterns in the literature. That means we need to create AI generated hypothesis as a starting point. Interesting, but always in need of rigorous experimental follow up. Responsible research design, transparency in how hypotheses are generated, and cross validation with biological data are all essential to avoid scientific dead end.
The concept of virtual patients, AI models that simulate individual responses to drug based on genetic, clinical, and imaging data, promises to revolutionize clinical trial designs. Yet each brain is unique in ways we don't actually understand. So, how realistic is the goal to accurate individual response prediction? Are we approaching personalized neuroscience or sophisticated guesswork?
The idea of virtual patients is so exciting, using AI to predict how a specific person might respond to a treatment based on genetic and clinical data. But the reality is, every brain is unique in ways we are only beginning to map out. Right now, these models give us sophisticated estimates rather than exact predictions. They are already making clinical trial designs more efficient, identifying who's more likely to benefit from a drug. But we are still at a frontier between personalized neuroscience and educated guesswork.
Right, so graph neural networks can model the brain's connectome and predict how drugs might propagate through neural circuits. This could revolutionize our understanding of network pharmacology. However, these models require very detailed connectivity data that varies between individuals and changes over time. How do we build reliable network based drug discovery platforms on such dynamic foundations?
Graph neural networks are compelling because they can, in theory, model how drugs move through brain circuits rather than just acting on single targets. The challenge is our data. Brain connectivity varies from person to person and can change with age, illness, or even the time of a day. Building reliable platforms will mean integrating dynamic, multi model data from imaging, genetics, and longitudinal studies, and accounting for uncertainty. We are not there yet, but it's a promising decision for future drug discovery.
Right. So, thank you, Jhansi, for joining us in our podcast, Neural Frontiers, and thank you, listeners. If you like our podcast, do not forget to like, share, and subscribe. Thank you.